Okay, so welcome back to this next video in which we are discussing the mammalian circadian timing system. Okay, so we have just been discussing the suprachiasmatic nuclei, okay, and we've been discussing the position of the suprachiasmatic nuclei within the hypothalamus. I just want to draw you one last little picture to show completely where their position is, and this is going to be a picture where we look from the front this time. Okay, so what I'm going to draw is the optic chiasm then. I think I'll draw it slightly lower down than that. So we'll have the optic chiasm here, okay, and into the optic chiasm, of course, will be coming the two optic nerves, okay. So this will be the left optic nerve here, coming from the left eye, okay, and on the other side you'll have the right optic nerve, so we'll colour in the left and right optic nerves in red here. Okay, so there are the optic nerves. Then they are exchanging in the optic chiasm here, which is in blue. Okay, and then what will then be leaving the optic chiasm is then the two optic tracts. Okay, so then coming out you'll have the two optic tracts here, one there and one there. Okay, so this again is the left optic tract, and of course what happens in the optic chiasm is you exchange fibers which are conveying information about the opposite sides of the visual field. Okay, so I'll just have a little discussion of this in a moment. Okay, so if I draw very basically the two eyes here, okay, so this can represent your left-hand eye and this can represent your right-hand eye, then basically we can divide both eyes in half. Okay, these sides, which I'll highlight in green, this portion of the left-hand eye and this portion of the right-hand eye are receiving information from over here. Okay, so from the right portion of the visual field. Okay, so this is the right visual field. Whereas these portions of the eyes, this portion in purple here and this portion in purple here, they're receiving information from over here, which is the left side of the visual field. Okay, so what will be happening is in the optic nerve you've got fibers conveying information about the right side of the visual field and the left side of the visual field. The idea is to pass all of the fibers which are conveying information about the right side of the visual field onto the right optic tract over here. So this portion here will be carrying fibers from the right side of the visual field. Okay, and you then want all of the information coming from the left visual field to go down the right visual tract. Okay, so what will happen is the fibers from the right-hand eye here that are conveying information about the left visual field, they will remain on the same side. They won't go through the optic chiasm. Okay. Meanwhile, the fibers from the right eye which are conveying information about the right visual field, those will go through the optic chiasm to the left optic tract. Okay. Now let's do the same thing on the left eye. The fibers which are conveying information about the left visual field, those will go through the optic chiasm and then go into the right optic tract. And then finally, the fibers in the left optic nerve which are conveying information about the right visual field rather, they will remain on the same side and they will go into the left optic tract. Okay, so that's just a little bit of a revision of what the optic chiasm is all about. Okay, so we have our optic tracts then now in turquoise here. Okay, now the hypothalamus then is sitting here. Okay, so we've got the front of the hypothalamus sitting here, and we've got the two suprachiasmatic nuclei pretty much perched like so. Okay, with the third ventricle sitting in between those two suprachiasmatic nuclei here. Okay, but of course you can't see that because there is this portion that is in front of the third ventricle here. So that's why I put it in dashed lines. So they are separated by the third ventricle, but they're sitting just above the optic chiasm, like so. Okay, so that's why they're called the suprachiasmatic nuclei. Let's now have a look at the structure of these suprachiasmatic nuclei in a little bit more detail then. So basically they're divided up into two portions. Okay, they're divided up like so. Okay, now I'll pull one of these out. I'll pull out the left suprachiasmatic nucleus here. And let's draw a bigger picture of that division. So here is the entire thing. And then we divide it up like so. There is this portion here, which is known as the dorsomedial shell. Okay, so this is called the dorsomedial shell of the suprachiasmatic nucleus. Okay, and you will notice that it faces the midline, so that explains why it's the medial portion. Medial means that you face the midline, so it's closer to the midline of the brain, or the midline of the entire body, in fact, than is this portion down here. But why is it called dorsal? Okay, it's surely superior, it's towards the top, it's above this portion, so why is it towards the back? It's not towards the back, but then you have to remember the way that we use ventral and dorsal with regards to the brain. Okay, so if I draw you again this picture of the cerebral hemisphere here, okay, like so, basically dorsal means towards the top of the brain. Okay, so you have to imagine that the brain is effectively this portion that has been flapped down. Okay, the spinal cord is nicely vertical like that, and it's as though the brain was a portion that should be facing upwards like this, and it's been flapped down. Okay, so we call this wait, what if I put a shell oh no, that's sorry, yes, ignore that. It looks as though I've labelled this shell now, but no, this is the dorsal portion, basically, the dorsal aspect. So things that are towards the top of the cerebral hemispheres, we call those dorsal structures. And that explains why this bit's called the dorsomedial, because it's towards the top of the brain, but we call that the dorsal aspect. Okay, towards the bottom, we call that the ventral aspect. Okay, so usually dorsal and ventral mean front and back. So what do we now use for the front and the back then of the brain? Basically, towards the front we call rostral. Okay, and that strictly means towards the nose, basically, which of course it is. The nose will be somewhere down here. Okay, and towards the back we call that caudal. Now caudal strictly means towards the tail, and of course if we had a tail it would be going towards where we are. Look at monkeys for an example. Okay, right, so this portion then is called the dorsomedial shell of the suprachiasmatic nucleus, and we can understand why that is called that now, because it is dorsally, when we view dorsal in terms of the way that they refer to the portions of the brain, and it is medially, certainly it's facing towards the midline. Okay, then this other portion that I'm now highlighting in light green here, this is what's known as the ventrolateral core of the suprachiasmatic nucleus. So this is the ventro, and then it should be all one word, but I haven't got enough space there. Ventrolateral core of the suprachiasmatic nucleus. Okay, so we further divide the suprachiasmatic nuclei, each of them, into these two portions. We have a shell and then a core, and the shell is dorsomedial, meaning that it's towards the back of the brain, which is the top, and it's also towards the midline, and this portion, which is towards the bottom of the brain, therefore ventro, and away from the midline, lateral, that is called the core. So this is the ventrolateral core. Okay, right, so that finishes our anatomy of the suprachiasmatic nucleus. Let's now talk about how they are the master clock. Okay, so basically the suprachiasmatic nuclei, both of them, consists of a huge number of neurons. Okay, so it's this collection of neurons, and all of these neurons have within them, like all cells in the human body, they have circadian oscillators. And all of these neurons in the suprachiasmatic nuclei, their circadian oscillators are perfectly in phase. So all of the neurons are oscillating in phase. Now, how do they do this? Well, they all have neural connections with one another, so that may be the reason. In addition, they're all secreting molecules that all of the others can feel and sense. Okay, so they are all oscillating perfectly in synchrony with one another. Not only that, but they are also oscillating in synchrony with the light-dark cycle. So let me explain what I mean by that. Okay, so we'll draw these graphs. It really is the only way to get this across. Okay, so we'll have on the y-axis light. So we've already drawn this graph. It was the first one that we drew. Okay, and then on the x-axis we'll have time, so varying between 0 and 24 hours. And of course, the light-dark cycle is that the level of light goes up during the day, and then oh dear, I've overshot completely again. Let's move this. Let's move it over to here. 24. I should draw the graph and then label up the axes. Okay, so it's coming back down to here. Okay, so there we have time, 24 hours. And of course then it repeats over and over again every day. Okay, so this is the light-dark cycle. This is the master Zeitgeber, as it's called. Okay, so this is an external oscillator that we want our oscillators inside the body to oscillate with effectively. Zeitgeber just means time giver in German. Zeit, I believe, is time in German, and then Geber is the verb to give or whatever. So time giver. Okay, so it's something by which we can oscillate in time to, basically. Okay, so all of the suprachiasmatic nuclei, not only are all of their internal clocks oscillating in phase, so the levels of period in all of them will be roughly the same at every single time that you look, okay, but also they are oscillating beautifully in phase with the light-dark cycle, so their period levels will go up during the day and down during the night, and I was trying to get them completely beautifully in phase, but never mind. Okay, so the circadian oscillators within these neurons, within the suprachiasmatic nuclei, oscillate in phase with the external light-dark cycle, basically. Okay, and this structure, these suprachiasmatic nuclei, they are responsible for making sure that all the other cells of the body are also not only oscillating in synchrony, but also oscillating in synchrony with the light-dark cycle. So we want all of the cells in the human body to be at the same phase in this cycle of period and cryptochrome at the same time, basically. We don't want them all having their own internal times. We want them all to believe that the time is the same, basically. So we want them all to be set at the same time, okay? And we also want that to be correct according to external time, basically, according to the external Zeitgeber of the light-dark cycle. Okay, and it's the suprachiasmatic nuclei which are responsible for this. How do we know this? Well, basically, if you remove the suprachiasmatic nuclei from an animal, then all of the cells within the body lose their synchrony. Okay, they start oscillating completely out of phase. So all of the cells of your body effectively think that the time is totally different from every other cell, basically. So it's chaos. They all think it's a different time. Okay, so if you remove the suprachiasmatic nuclei, you lose synchrony. You don't lose the circadian oscillators. All the cells continue having their little circadian oscillators, but they all become out of synchrony, basically. So synchrony is lost if you remove the suprachiasmatic nuclei. Even better, if you transplant suprachiasmatic nuclei back into this poor animal that's had its suprachiasmatic nuclei removed, okay? If you put a transplant of fetal suprachiasmatic nuclei in, and you use fetal suprachiasmatic nuclei because that has a better chance of actually regenerating and integrating into the original animal's brain. Okay, so you take fetal suprachiasmatic nuclei tissue, which will still have the ability to actually divide, you put that back in and it restores the synchrony within a few days. Okay, so restoration of synchrony. So all of the cells start oscillating again in synchrony, basically. Okay, so that's very convincing that then the suprachiasmatic nucleus, or nuclei rather, are responsible for synchronizing all of the internal clocks within all of the cells of our body. Okay, even better, what you can do is an experiment where you transplant the suprachiasmatic nuclei back in, but you don't do it, you don't just put them in. Instead, what you do is you wrap them in a certain porous plastic. Okay, so what you'll do is when you transplant in the fetal suprachiasmatic nuclei material, okay, so let's say in orange here, this is the lump of fetal suprachiasmatic nucleus material. What you're going to do is you're going to wrap it in a type of plastic shown in blue here. Okay, and this will stop the suprachiasmatic nucleus material from forming neural attachments with other neurons within the brain. Okay, so you stop it from actually integrating into the brain, but the plastic is porous and allows hormonal molecules, okay, so signaling molecules to leave it. So the neurons can't make connections, but this can release signaling molecules. So it's a porous plastic coat, okay, and you might think that this would fail to restore synchrony of the cells of the human body, because you might think what has to happen is the suprachiasmatic nucleus has to form neural attachments with everything else in the brain, and then it must send out, loads of neural signals to all the cells of the body and get them to synchronize together, okay? But what you find is that if you do this, it still restores the synchrony, which tells us what? It tells us that the suprachiasmatic nuclei are releasing some sort of molecule that is telling all the other cells of the body what time their clock should be, and therefore setting the phase of all of the circadian oscillators within all of the cells of the human body. Okay, so unfortunately we do not know what this molecule is that is telling all the other cells of the body what time it is effectively. There are three main candidates that we have, however, which are released by the suprachiasmatic nuclei. Okay, so the first is TGF-beta, standing for transforming growth factor beta. Okay, so here it is. So this is a protein, okay, which the suprachiasmatic nuclei neurons do produce. Okay, and it stands as a candidate for the molecule that tells all the other cells of the body what time it is. Another candidate that we have is something called prokineticin 2, okay, usually abbreviated to PK2. So this stands for prokineticin, okay, and then 2. So the P is obviously for pro, and then the K for kineticin, and then the 2 for 2. Okay, and the final one is something known as CLC, which stands for cardiotrophin-like cytokine. Okay, so the first C is for cardiotrophin, then the L is for like, and then the C is for the second C is for cytokine. Okay, so all of these molecules stand as potential molecules that could be involved in this signaling from the suprachiasmatic nuclei to the other cells of the body to tell them what time it is. Okay, so overall, where have we got to now? Well, we have this master clock. Well, we have two of them within the brain, the suprachiasmatic nuclei, which are oscillating in phase with the light-dark cycle of the external world. Okay, and these are responsible for sending signals which we think are diffusible molecular molecules, okay, which will go out to all the other tissues of the body and tell them what time their clock should be set to, so that all the other cells of the body are oscillating in phase with the suprachiasmatic nucleus, and then therefore in phase with the light-dark cycle of the external world. What we want to turn our attention to in the next video is how the light-dark cycle of the external world entrains the suprachiasmatic nuclei, which means how do the suprachiasmatic nuclei get their clocks in time with the light-dark cycle. Okay, so they must be receiving information from the eyes, basically, and indeed they are, and we'll look at that in the next video.